All right, today we're going to cover meiosis, which is very similar to mitosis. If you understand mitosis and you have the phases memorized and you're familiar with the vocabulary terms that we discussed, meiosis will be well, it should be okay, I guess. I don't want to say it should be pretty easy. It's still kind of complicated but I do add a few more things in. So the foundation of this would be mitosis and the phases of mitosis. So let me just go back, let me go through some background information and then we'll get into what the steps are. So genes are going to be segments of DNA that code for basic units of heredity. And like we learned yesterday, DNA codes for RNA, RNA codes for proteins. And so your genes ultimately code for proteins, and proteins are the reason why you have your traits. Offspring acquire genes from parents by inheriting chromosomes. One set of chromosomes from mom, one set of chromosomes from dad. And so there's different types of reproduction. So when I say one set of chromosomes from mom and one set of chromosomes from dad, I'm actually talking about sexual reproduction. But there is a form of asexual reproduction in many organisms. There's not two parents, there's just one. So let's kind of go through the differences between asexual and sexual reproduction. So asexual reproduction is going to produce clones. That means they're genetically identical to the parent. There is a single parent. There's little variation in the population. If they're clones, then there's little variation. So if you think about it, your mom looks a certain way, your dad looks a certain way, you look different than either one of them. You're not an exact copy of either one of them. So there's a lot of variation in traits. But with asexual reproduction, it's going to be, the offspring is going to be an exact clone, so little variation. Asexual reproduction, though, is fast and energy efficient. And the examples are called budding. Budding is how yeast will reproduce, and then binary fission. So binary fission, you just have one bacteria cell. So under budding, if you can write yeast, like the yeast that you did that you used in your lab the other day. And then under binary fission, if you can write bacteria. All that is that a yeast cell will just kind of split apart, or a yeast, like a group of cells will just kind of split apart. A binary fission, it's cells. So budding would be a yeast cell that just kind of splits. Binary fission would be a bacterial cell and it just splits in half. Kind of like mitosis. It kind of looks like mitosis, but that's just how they reproduce. And it's usually unicellular organisms that can do that. And multicellular organisms, because yeast is a fungus. All right, sexual reproduction. Meiosis is the process that produces gametes called sex cells, sperm and egg. I would write sperm and egg next to that. There's two parents. One is a biological male, one is a biological female. There's lots of variation and diversity. It is slower and energy consumptive. In our example is humans, trees. Yes, plants reproduce sexually. That's why we need bees to get pollen and go and put it on another flower, and then it goes down the pollen tube into the place that has the eggs, and then it fertilizes an egg, and then it grows, and then petals fall off and you have a fruit. So yes, plants reproduce sexually. Here's something really, really interesting. I love this. Can a sexual reproducing organism become an asexually reproducing organism? Yes. Not all of them, but there are some. So you see I have a picture of sharks and I have a picture of bees. Sharks, if a female shark is put in captivity long enough, she can reproduce asexually. That's called parthenogenesis. So make sure you write down that word. So it can happen. It's not ideal, but in captivity, they have seen, the zookeepers or the aquarium keepers have seen female sharks on occasion, not every time, reproduce asexually. So what is the biological sex of their offspring? If sharks reproduce asexually, the biological sex of their offspring will be female. Now it's interesting with bees, it's different. All right, in bees, um, I mean, talking about bees and teaching about bees, obviously, if you know anything about me, you know I'm the bee person. I can spend a really long time talking about bees, but I'll just tell you that female worker bees can reproduce asexually. Can lay an egg. And what's interesting with bees is asexually reproducing female worker bees produce only male offspring. The queen bee, who goes out and sexually reproduces with male bees, can lay females and males. But so when you see in a hive all males, which no offense, male bees are really big and bulky and they're slow and they eat too much and they're just not good for much except for reproducing. And so in the winter, the females will kick all the males out. So if you look at it, if I go and do a hive inspection in our beehives on campus and I see all male eggs being laid, and there's a way to tell if they're male or female before they even hatch, I have to get really, really worried because I do not want an all male colony. They will just eat everything and not make anything because they are kind of lazy. So that means that the queen is not laying eggs right if it's an all male colony because it means the workers are laying and they're not supposed to do that. So anyway, that's a whole nother subject. Just so y'all know, sharks and bees can reproduce asexually, but when their offspring are not going to be as good, as viable, as strong and healthy as if they would have reproduced sexually. An interesting fact, the New Mexico whiptail males have become obsolete because of parthenogenesis, so the females are going to only produce females. There are no more males in the New Mexican whiptail, and it can't be reversed because there's no male chromosomes there anymore. So I thought that was kind of cool that happened, that it went to just an all-female group. I'm trying to get my notes up here. That's why I'm stalling a little bit. Sorry. So let's talk about body cells. So body cells we talked about before. Somatic body cells are anything that is not a sperm or an egg. In humans, you're going to have 46 chromosomes in a body cell. 23 from mom, 23 from dad. There's 2 sets. That's what that 2N means. 2N means there's 2 sets of chromosomes, one from mom, one from dad. There's 46 total, 23 from each parent, and it is a body cell. Each pair of homologous chromosomes includes 1 chromosome from each parent. So I'm going to show you a karyotype again in a minute so I can show you what homologous chromosomes means. That just means chromosome number 1, there's 2 of them, one from mom, one from dad. Those 2 are called homologous chromosomes. They come as a pair. And out of all of the homologous chromosomes, 22 are considered autosomes. They're chromosomes that don't determine sex. And then 1 pair is considered sex chromosomes, X and Y. I am going to go over this using a karyotype because I feel like when I just say it and you see it as a text, it's kind of confusing. So I want to go back over this using a karyotype in just a second. Sex chromosomes in humans are X and Y. For females, biological sex XX makes them a female. For males, XY is for biological sex. Now, I want to make sure that I say here, this does not have to do with gender. I'm talking about biological sex that a person is assigned at birth. Okay, so I want to make sure that everyone knows I'm not getting into any gender discussions here, just biological sex. All right, and then gametes. So if you have somatic cells or body cells that are not sex cells, autosomes or chromosomes that are not sex chromosomes, then you have your sex chromosomes, then you have your sex cells. So this is kind of confusing. I probably should rearrange this slide. Somatic cell and gametes kind of go together. One is a body cell, one's a sex cell. And then autosomes and sex chromosomes go together because they're both types of chromosomes. So the arrangement on here isn't very good. So just so you know, a body cell is a cell that's like a skin cell. It's a whole cell with all the organelles and everything. A gamete is a cell that is a sperm or an egg. An autosome is a chromosome found inside of a cell that codes for something that doesn't determine biological sex. And then sex chromosomes are your X and Y that do determine biological sex. So hopefully I didn't just confuse everyone really bad. So homologous chromosomes in a somatic cell. So this is a body cell. Let's say this is after S phase of interphase and cell division is beginning to occur. You have two sister chromatids here. You have two sister chromatids here. Because these are both the same length, those are considered homologous chromosomes. One came from dad, one came from mom, and they're with their copy before cell division. Okay? If you look here, and then here, these are the same length, so therefore those are the same chromosome. One came from dad, one came from mom. They have their sister chromatid attached to them because replication already occurred and they're about to divide. And then here you would match these up as homologous chromosomes, one from mom, one from dad, and they have their copy with them. Okay? So in meiosis, and then like I said, I'll go over the karyotype a little bit later and I'll go over all these vocab terms again, but I'm going to move on and talk about meiosis. Meiosis, the difference between meiosis and mitosis is in meiosis, the cell divides twice. Instead of resulting in 2 cells, this one results in 4. So here's your original. It divides once, then it divides again. And you end up with not 46 chromosomes, you end up with 23. Okay? So the first division during interphase, the chromosomes replicate just like before. In prophase 1, homologous chromosomes pair up. It's going to be different than in mitosis, and that's called synapsis. And so it forms a tetrad where 4 sister chromatids are together like that. That's not what you saw before, right? What you saw before is only one of these X's was there, but in meiosis, 2 X's pair up together, the one from dad and the one from mom that matches up. They're homologous, they match up. Okay? Crossing over occurs, which means this does not happen during mitosis, only in meiosis, which means these homologous chromosomes are going to start exchanging pieces. They're going to start exchanging pieces. That's called crossing over. And occurs at the chiasmata. The chiasmata is just the breaking point on the chromosome where they switch places. And that's what it looks like. Chiasma, chiasmata, same thing. All right. In metaphase 1, the tetrads, that's the 2 homologous chromosomes with their copies, line up in the middle. In anaphase 2, homologous chromosomes separate. Sister chromatids are still attached. The blue one goes to the left, the red one goes to the right, but the chromatids, their copies are still attached. And then telophase and cytokinesis, is the same as before. The nucleus doesn't really reform because there's another division, but you have an actual cell division like you did before. So in some species, the nucleus reforms. I can't even think off the top of my head which ones do that. So you can just cross that out because if I don't remember, I mean, I should probably look that up, but for the sake of time, we're just going to cross that out. The nucleus doesn't actually reform, and then there's another division. Okay, here's another picture to show you what's happening. So it's another picture to just show you what's happening in case you were confused. Prophase, chromatin condenses into chromosomes. Okay, they are going to exchange pieces. So one piece of one chromosome is going to get stuck on the other one. So remember, let's pretend this is chromosome number 1. Red is from mom, blue is from dad. Each chromatid, has their own copy with it, and then they start exchanging pieces. All right. I know this is kind of confusing. They line up in the middle, they get pulled apart. Do you see how this one isn't all red? There's some blue parts on it. Why is that? What you should answer is, oh, because of the crossing over that occurred. They exchanged pieces. I hope you can start kind of seeing why, even though you are the offspring of your mom and dad, and they're the offspring of their mom and dad, you're never going to be identical to anyone in your family. And it's because of this crossing over that occurs. All right, and then cleavage furrow, cytokinesis, it forms 2 cells. Okay. The second division, there's no interphase, there's no crossing over, a spindle forms, chromosomes line up, Sister chromatids separate, and then you end up with 4 cells at the end, and each of the daughter cells is genetically unique, not identical to each other, not identical to the original cell. All right, so I think you have just a couple of little blanks to fill in. All right, so here we go. This is meiosis 2. Prophase, chromosomes condense more. Metaphase, they line up. Anaphase, they get pulled apart. Telophase and cytokinesis, there is a division. All right. So events that are unique to meiosis 1 that don't occur during mitosis. So what is the difference? What are the differences? So the differences are synapsis and crossing over. So crossing over doesn't happen during mitosis. In meiosis, you have homologous pairs lining up, where in mitosis, they individually were on their own spindle. And then during anaphase 1, you still have sister chromatids attached to each other, and that doesn't happen during mitosis. All right? Okay, so how does all of this relate to sexual reproduction? And so what we're going to do is look at a life cycle, which is the reproductive history of an organism from conception to the production of their own offspring. So fertilization and meiosis kind of go hand in hand because they alternate in the sexual life cycles. Meiosis is the cell division that reduces the number of chromosomes from 46 to 23, creates sperm and eggs. And then fertilization is the combination of the sperm and egg to make what's called a diploid or diploid organism that now again has 46 chromosomes. A fertilized egg is called a zygote, and that 2N represents it has 2 sets of chromosomes, 1 from each parent, so a total of 46. It's known as diploid, and I don't think I put that word in there. If you guys can, actually it's in the next picture, so I'll have you highlighted in the next picture. Then the zygote divides by mitosis to make a multicellular organism. So I'm going to go through a picture in a second that might help with all of these words. So pause right now if you need time to write down the rest of the words. I'm going to move on. All right, so here's a picture to go with all of the text that you just looked at. So you have a man and a woman. Inside his testes, meiosis is about to happen. Inside her ovaries, meiosis has actually already happened before she was ever born. So meiosis occurs when a 46 chromosome germ cell divides by meiosis, divides twice to make an egg. In the testes, it's a 46 chromosome germ cell that divides twice to make a sperm. So the egg has 23 chromosomes. The sperm has 23 chromosomes. That's called haploid. It's only one set of chromosomes. It's 23. It's called haploid. I need you to circle that word. Circle that word. Then, through fertilization, the sperm and the egg unite to form a diploid or diploid zygote. So do you see how the 23 chromosomes of the egg from mom combine with the 23 chromosomes from the sperm from dad to form a diploid 46 chromosome cell? Then that cell divides by mitosis to eventually make the adult. And then inside the adult, you have germ cells that divide by meiosis to make sperm and eggs, and then sperm and eggs unite. So you can see it's mitosis and meiosis, that is the human life cycle. Hopefully that made a little bit of sense, I hope. So we'll talk about a little bit about genetic variation. So remember I said that you are not an exact replica of anyone because of crossing over. So genetic variation just means a variation in genetics. Don't end up with clones, right? This is a really, really nice picture that shows a very simplified version of meiosis. And so you have your homologous chromosomes, crossing over occurs, there they are, they switch places, one division and then a second division. Do you notice that let's pretend that these are sperm. None of these sperm are identical to each other at all. They're very unique. And so that's considered genetic variation. So crossing over causes a lot of genetic variation because of the exchange of genetic material between homologous chromosomes, and then they're called recombinant chromosomes. They're recombined or something like that. So these would be considered recombinant chromosomes right here. Independent assortment of chromosomes. So remember, homologous chromosomes line up on the metaphase plate right here in the middle during metaphase. They could line up like this, or they could line up like that. And do you see how if they lined up like this, it would result in a difference than if they lined up like this? So that can result in genetic variation depending on how the homologous chromosomes line up. That's called independent assortment of chromosomes. Okay, so there's two possibilities. There's a 50-50 chance. So random orientation of homologous pairs in metaphase 1. And remember, red is maternal, blue is paternal. And so the maternal chromosome is right here, the paternal chromosome is right here. So do you see this gamete is going to have a combination of mom and dad? This gamete is going to have mostly dad. So that's why when you see kids, they can look more like one parent than the other. It depends on how the chromosomes lined up, basically. Or they can look like a grandparent or something like that. And then another source of genetic variation is going to be random fertilization. Any sperm and any egg, 8 million sperm. So if a person has 8 million sperm and a man has 8 million sperm, a woman has 8 million eggs, there's a 1 in 64 trillion, there's 64 trillion combinations. So there's a 1 in 64 trillion chance that you came to be. You are so special. All right. This is showing you, once again, differences between mitosis and meiosis. It's a really important picture to look at because remember when you made your stop motion video, you only put 1X, right? Kind of lined it like that. And then they were identical. And you only ended up with 2 cells and they were identical. With meiosis, remember we're making sperm and eggs here, so it's different. You end up with, you have crossing over, you have homologous pairs lining up together instead of individually, and you have 2 divisions, and each of the 4 daughter cells look different and unique. They're not clones. All right, so those are the differences. And then the other main difference is in mitosis, the resulting daughter cells have 46 chromosomes. In meiosis, the resulting daughter cells each have 23. And then this is a chart. So you're going to need to write this down. So they both have in common that they're both cell division. They're both divisions of a cell's nucleus, and DNA has to replicate before each one occurs. Mitosis is the division of somatic cells, and they're clones of each other. Meiosis is the division that forms gametes. Mitosis has 1 division, meiosis has 2. Mitosis ends up with 2 daughter cells, meiosis ends up with 4. Mitosis, the daughter cells have 46 chromosomes each. Meiosis, the daughter cells have 23. So diploid, haploid. You're probably going to have to pause. I'm going to have to pause. Mitosis, they're clones. Meiosis, they're genetically different because of crossing over. Mitosis occurs from zygote to death. So both males and females have mitosis happening from birth to death, to repair tissue, to grow. And then meiosis happens in females before birth. Birth follicles are formed and mature ova are released beginning in puberty. So basically your eggs are made before you're even born. So you're gonna have to pause if you need to do this. If you still need to fill this in, I'm gonna move on. Oh, just kidding. Just kidding, there was a couple more. So the purpose of mitosis for growth and repair, the purpose for meiosis is reproduction, and then there's no crossing over in mitosis. This is going to take some looking at. This is probably going to take some studying. So you did your stop motion video and that forced you to really look at the mitosis. You're going to do the same thing with meiosis, except you're using your candy to do it. And this time it doesn't have to be a stop motion video. It can if you want it to, but this time you just take pictures and you can put them on a document going in order. All right, so let's talk a little bit about karyotypes. Karyotypes, I need to adjust myself. Sorry if it's making a weird noise. Karyotypes are a picture of a complete set of chromosomes arranged from the largest to the smallest pair. So I want to start throwing around some vocab words that you've learned. Okay. This is not a chromosome and its copy. These are homologous chromosomes before DNA replication occurs. So what would a karyotype actually look like right before cell division? How many actual chromosomes would you have as a human if you had a karyotype of your cells right before division? How many would you have? Would you take a guess? Right. You would have 90, hold on, 92. You would have 46 plus 46, 92 on here. So that's something important to remember that will help you kind of conceptualize. Oh, so these are homologous chromosomes. It's chromosome number 1. They have the same kind of genes. So let's say eye color is on chromosome 1. The protein that codes for eyesight is on chromosome number 2. The protein that codes for what my nose is going to look like is on chromosome number 3. And I'm just totally like making random examples here. But each chromosome is going to code for very specific things. Chromosome number 1, you have one from mom, one from dad. Those are homologous chromosomes, but the DNA is going to copy right before cell division. So you would actually see the tetrad right here of the 4. All right. So if I could color code these, I'd make one red, one blue, one red, one blue, one red, one blue. And then right before cell division, you'd have 2 red, 2 blue, 2 red, 2 blue, 2 red, 2 blue. Hopefully that kind of made sense. So these are homologous chromosomes because they are both the same type of chromosome. They have the same type of genes on them. One's from mom, one's from dad. Okay. There are 22 autosomes. Remember what that meant? 22 autosomes. That's this, 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 this one, 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 1 to 22. They're autosomes. That means they have genes on them that don't determine anything about biological sex and sex characteristics. They are genes for just proteins that are just proteins no matter if you're male or female. All right? Those are autosomes. Sex chromosomes are right here, the X and the Y. So this would be a biological male. Those are sex chromosomes. Okay, so hopefully that helped a little bit. That's karyotype. So if you were to make a karyotype, and there are virtual activities where you actually end up with this, and you have to match all the chromosomes up. I've had students do that before, but we don't really have a lot of time to do that in here because I want you to do your candy activity. But if I was giving you more work, I would give you a virtual activity where you were to drag chromosomes and match them up. So you'd match up homologous chromosomes. And this is how they actually do it. They stain them and then they match them up and they make a karyotype. Okay, and it looks like this. So there's 22 pairs of autosomes, chromosomes that don't code for sex characteristics, and 1 pair of sex chromosomes. Is this one a male or a female? Yep, it's a female because there's 2 X chromosomes. And then everything else looks pretty normal. Male or female? Yep, that's a male because it's X and Y. Everything else looks pretty normal. There's no extras or anything. In cancer, this is what a cancer karyotype would look like. Look at all that. It's just totally screwed up. I mean, look, you're only supposed to have 2 of chromosome number 2, but they have 1, 2, 3, 4, 5, 6. So a lot of mistakes were made that made duplicate chromosomes. This is a breast cancer cell karyotype. It's crazy, right? Looks super crazy. So you can see part of what happens in cancer is multiple sets of chromosomes, more sets of chromosomes than you need. And I'm going to talk about in a minute how that happens. This is a fluorescently stained HeLa cell. So these are one of Henrietta Lacks cells right here. It's one of her cancer cells, what they look like. Do you see how she's completely missing chromosome 3 in her cancer cells? This is not what all of her cells look like. This is what her cancer cells look like. So a bunch of mistakes are getting made. All right. Okay. So there is something called chromosomal disorders. It's a genetic disorder that is caused by non-disjunction, which is a failure for a chromosome to properly separate during anaphase of meiosis. That is very important to write down. A failure for chromosomes to properly separate during anaphase of meiosis. In cancer, it's more mitosis because the cancer cells arise spontaneously due to genetic mutations. And so these ones I want you to know, these happen during meiosis. So these are 4 disorders where chromosomes do not properly separate during anaphase of meiosis, and therefore a sperm or an egg ends up with an extra or a missing chromosome. So if we look back at our meiosis picture, sorry, I'm gonna have to go back here. I want to go back to the actual picture. Y'all know I do this. I'd like to go. Okay, right here. Anaphase. Do you see how these are separating from each other? Wouldn't it be a problem if this blue one went with the red one over to the other side? That's non-disjunction. All right. Same thing in anaphase 2. And it's most likely going to happen during anaphase 2 if it's going to happen. If it happens during anaphase 1, that's way too drastic, and usually a miscarriage would happen with that offspring. But here, look at this right here. Look where my cursor is. What if this red one right here actually went with the other red one? Do you see how this cell then would end up with a missing chromosome? And then this gamete, sperm or egg, would actually end up with an extra chromosome. All right, that's called non-disjunction, a failure for chromosomes or chromatids to properly separate from each other during anaphase. All right, hopefully that made sense. Okay, now I'm going to go back, all the way to the disorders. Okay, now this is the interesting part. Trisomy 21, a failure of chromosomes to separate during anaphase of meiosis, where a gamete ended up with an extra chromosome 21. Extra chromosome 21. So that's what I want you to write under trisomy 21. Trisomy means there are 3 chromosomes where there are only supposed to be 2, and it is chromosome 21. So if you looked at a karyotype of a person with Down syndrome, you would see an extra chromosome right here, a 3rd chromosome 21, and that just totally offsets all the balance. There's too many chromosomes on chromosome 21 being expressed, and it just messes a lot of things up. So you'd see an extra one right here. Okay. Congenital heart defects, gastrointestinal abnormalities, eye and ear problems, and obviously cognitive issues, right? A person usually will not make it past their 30s. Although I knew someone with Down syndrome that passed away. He was in his 20s. And then I know someone who is in her 50s who has pretty severe Down syndrome. She is still alive. That's awesome. Klinefelter's disease. Klinefelter's disease, I talked about a little bit. I showed you. This is a non-disjunction disorder where a person ends up with a male. A biological male actually has an extra X chromosome. So if you looked at the karyotype of someone with Klinefelter's, they would have an X and a Y, but then they'd have an extra X right here. So it's like they're both male and female. They're both like biologically male and biologically female at the same time. And so they'd have an extra X right there. So that's how they would determine if a person had Klinefelter's. It requires a medical diagnosis, obviously. Males are born, they're considered biological males because when they come out, they do have a penis when they come out. So, and I am allowed to say that as a biology teacher. And so a male born with Klinefelter's may have low testosterone, reduced muscle mass, reduced facial hair, reduced body hair. Most males will produce little or no sperm, even though they have the anatomical features of a male. They most likely will not be fertile. A lot of times they will have ADHD that accompany everything else. They'll have breast enlargement, delayed puberty, flaccid muscles, infertility, low set ears, osteoporosis, reduced sex drive, and a small penis. So this is what a healthy male would look like. This is what a male with Klinefelter's would look like. So Turner syndrome. Turner syndrome means it's a female who is missing an X. So that means something happened where that egg was supposed to get an X chromosome, but the egg shows up empty, or the sperm shows up empty with no X chromosome. All right. So a person with Turner's would end up with just, so they are considered female because they look female when they come out. They don't have a penis. And so it would just be, just pretend like this one isn't here. So she would just have one X chromosome. And so what does that do? Let's discuss that. Let's look at that. Turner syndrome results from a missing or incomplete sex chromosome. So she only has one X. Symptoms include short stature, delayed puberty, infertility, heart defects, certain learning disabilities. And so it makes you wonder, okay, X chromosomes help determine sex, but they definitely have genes for other things too. They have something to do with cardiac function, something to do with learning and attention span. So it's kind of interesting how these sex chromosomes actually have genes that code for things that actually aren't sex related also. But they're not considered autosomes because they do have genes that code for sex characteristics. So treatment for Turner syndrome involve hormone therapy, and there is fertility treatment, and women with Turner syndrome actually can become pregnant, can have babies. It's just really, really hard, and they have to get a lot of fertility treatment. So this is what a person with Turner syndrome would look like. This person is actually, I think she's in her late teens, and so you can see how short they are, and these are all of the other features that they would have. So kind of interesting. Ooh, this one's my favorite. I don't know why this one's my favorite. Maybe it's because my son's named Jacob, and he seems to have a lot of testosterone. But I'm pretty sure he doesn't have this. Jacob syndrome, I actually think it's interesting because of the social implications of it. Well, they all have social implications to a certain degree. But this is a male. And I'll tell you also another reason why I find it interesting. This is a male that has an extra Y chromosome. So his karyotype would look like this, but he'd have an extra Y there. Jacob syndrome is also called super male syndrome. And I think that's one of the reasons why I find this one so interesting is that it's interesting because it almost seems like something that a male would want to have, a super male, like a lot of testosterone, really tall, lots of muscle tissue. But it ends up leading to a lot of aggression, and it has been found that some males are more likely to end up incarcerated if they have this syndrome right here. But they are unable to use this as a disability at this point in time. All right, so that is all on meiosis. Write down any questions that you have or any confusion you might have, and we will discuss that in the Zoom tomorrow. I'll talk to you guys later.